After 5 years, I still love RNAi, and here's why. You might think that I get bored of this thing that I study called RNA interference or RNAi, but I don't. Even when I spend all day on the weekend trying to figure out how to introduce this topic in a more general way as I'm trying to prepare for my thesis defense, where we have to give this more public presentation to family and friends and stuff, and so I'm trying to describe it in a more general way. And so today I was just working on it, and this is my first draft, and it needs a lot of help. But I thought I would share, in case people had thoughts or suggestions, things that confuse them, don't confuse them, whatever. So let's go. First, I realized I was too exhausted and forgot the cape. Sorry guys. Here we go. Proteins are held in the form of genes, which are stretches of DNA. Different proteins have different genes, and your genome has all the protein recipe. The instructions for making every protein that your body will ever need are held in stretches of DNA called genes. So different proteins have different genes, and the cells regulate what they want to make, when, and how much. So your cells don't go directly from gene to protein. Instead, they go through an intermediary called messenger RNA or mRNA. This mRNA is a copy of the gene, an RNA copy of the gene that gets made in this process called transcription. Then in a process called translation, a protein-making complex called a ribosome travels along the mRNA and uses it as instructions for making the corresponding protein. These mRNAs can be used multiple times, and you can have multiple ribosomes on the mRNA at the same time. But ultimately, the amount of protein that's made corresponds largely to the amount of mRNA that's present. But cells can change the amount of mRNA that's present in order to change the level of protein in the cells. So one way to do this is at the transcriptional level, by making more of the mRNA or by stopping making it. But this is really good for big changes, but it takes a while to take effect, and even if you were to stop the transcription at the source, so you're not making more mRNAs, but you still have all those mRNAs that are already there. And so what if cells want to deal with those? I study a way that allows the cells to regulate the levels of these mRNAs after they're made, that is post-transcriptionally. And this way is called RNA interference or RNAi. So it can actually cause the specific mRNAs to get degraded and prevent ribosomes from using them, leading to less protein being made. So it doesn't actually act on the protein directly, but it prevents more protein from being made. And then that protein that's already made can get degraded in other processes. So this is really great for helping control the levels of protein, and it acts faster than transcriptional regulation. But the really, really cool thing about this is that it's sequence specific. So it targets specific messenger RNAs to reduce the levels of specific proteins. So it's really important for our cells to use it. And it's also an important therapeutic tool because, as I'll show you, it can easily be programmed. So at the heart of it is this protein that I study called Argonaute or AGO. AGO is pretty cool on its own, but it gives us real power when it binds to a small RNA to form a core RNA-induced silencing complex or RISC. So these small RNAs can be microRNAs or siRNAs, and I'll tell you more about those later, but for now, just know that they're around 22 nucleotides long. And at their 5' end, they contain the 6 to 8 nucleotide long seed sequence that allows AGO to use them as guides to find and bind messenger RNA targets that contain complementarity to that seed, and often some additional complementarity. Then upon binding is when those translational repression and mRNA degradation can take place so that less protein is made. And this core risk complex, so AGO plus guide, it's incredibly stable. It has this half-life of weeks. So once AGO releases a target, it can then do it again to other messenger RNAs, but only those that have a corresponding sequence to the seed. So these could be in the same, like copies of the same messenger RNA or for different proteins, but they have to have those same sites. So different mRNAs can have binding sites for different microRNAs, and then the cell can control which microRNAs it makes so that AGO gets programmed in different ways to target different things. And experimentally, if we add in small RNAs to like cells, we can knock down the amounts of protein that are made. But AGO in our cells normally does it to more like modulate levels, so just kind of reduce levels a little and keep them stable in a nice way, like a thermostat kind of, instead of just like totally stopping the expression of the protein. But the way that it is able to control these, what it does is by controlling the levels of these microRNAs that it makes. And so let's talk a little more about what these microRNAs are. So microRNAs are what we call non-coding RNAs or functional RNAs. So they have a function as RNAs. They don't code for protein, so they don't have protein making instructions. But they still come from genes. They get transcribed just like those messenger RNAs we were talking about, but here they don't get made into messenger RNAs. Instead, what they get transcribed as are these long precursor hairpins called primary microRNAs. So this is taking place in the nucleus, which is the membrane-bound compartment of your cells where all of the DNA is. And once it gets transcribed, it gets cleaved by this microprocessor complex. So it's this protein complex that you can think of as a pair of like RNA scissors. And so it's going to cut off the duplex, this long hairpin, and then it gets shipped out of the nucleus into the cytoplasm, so the general cellular interior. And there it's going to meet another pair of RNA scissors called dicer, this protein called dicer. And this is also where like exogenous, so outside sources of double-stranded RNA can come in. And this can serve as precursors for small interfering RNA or siRNA. So siRNA is used a lot by insects and other invertebrates and plants as an antiviral defense mechanism. But our bodies have evolved more like complex immune systems, so we don't use our RNAi machinery in this purpose. But we can still add siRNA artificially in order to target specific messenger RNAs and like knock out or knock down proteins, and so this or the mRNAs. And so this is really important experimental tool and part of the reason why people have been so interested in studying this process. But so siRNA is going to come in so if you have long double-stranded RNA, that can come in here and be processed by dicer. And what dicer is going to do, it's like a molecular ruler and scissors in one, and it's going to chop it to this RNA duplex. So if you like buy siRNA, it's often what you're buying is like an RNA duplex, and then this gets loaded into Argonaute, and then Argonaute has to release one strand, the passenger strand, so that it exposes that seed region so that it can go find the targets to bind. And then once it finds a target to bind, what happens depends in part on the Argonaute and the part on the amount of complementarity between that guide and that target. So humans have 4 Argonaute proteins, and our main Argonaute protein, AGO2, has this like slicing ability. So what this means is that if that target is fully complementary, AGO can cut the target, which we call slicing, and then those ends can get chewed away by exonucleases, so you get mRNA degradation. So that's the case with like siRNAs where you have that perfect complementarity. But with mammalian microRNAs, you only have like partial complementarity between that target and that guide and that target. And so ego can't cleave it, and instead it recruits these cofactors. So it binds to this long scaffolding protein of the GW182 family, and it recruits the adenylation and decapping complexes that remove those protective ends from the messenger RNA so that they can get chewed away. And it can also cause that like translational inhibition, so keep those ribosomes from using it, and sequestration, so holding these mRNAs. And so these target sites, by the way, are typically located in the 3' UTR, so the 3' untranslated region. So if you look at like a messenger RNA, it's actually like this is not the scale at all, but it has this long open reading frame, which is the part that actually has the protein making instructions. And then it gets this like cap and this poly-A tail. But then between that tail and the end of the protein making instructions is a bunch of regulatory information. So it's the part, this is where the binding sites for the microRNAs usually are. And so this poly-A tail is generic and this cap is generic, but this 3' UTR is specific for this protein. So it's like part of the gene that gets made. It just doesn't have it's not translated. It doesn't have the protein making instructions. It has the regulation for the protein making instructions. But basically, as an end result of these, you get target repression, and then ego can release the target and do this again and again because it's super awesome like that. And so depending on what microRNA it's programmed with or which siRNA it's programmed with, it can target different things, and then you can have different combinatorial effects from having mRNAs with different combinations of microRNA sites and different mRNAs with the same microRNA site as other messenger RNAs. And so this really allows for all this combinatorial and this like regulatory stuff, and it's all just really awesome. And what's really, really awesome is the protein at the heart of it all, ego. And so that's what I study, and why.